Hello everybody. Today we're going to talk about the basics of genetics. We start with the initial stage from where genetics started, and that is talking about the person who started it, Gregor Johann Mendel. Do you know who Gregor Johann Mendel was? He was a priest. He was a priest in a monastery in Brunn. See, a priest in the monastery in Brunn, and he is credited with the discovery of the laws of genetics. However, his work was not recognized for a long time. For almost 34 years, he was not given the credit of finding these basic fundamental laws of genetics. You must have studied these in class 10. But in class 12, we are going to actually study them in detail. Not only in detail, we will talk about the history. We're going to talk about how these laws came about and how Gregor Johann Mendel started with the whole formulation of these ideas. What led to it? Alright, let's go into the basics of it. So, here was this man, he was a priest and he was talking about, different, he was a pretty good student. He went to Vienna for many different courses, and finally comes back to his monastery, and he starts doing experiments on pea plants. Alright, and pea plants are known as Pisum sativum. See, Pisum sativum is the scientific name for pea plant, the common garden pea. He found two different types of garden pea in his monastery. So he decided, alright, why not? Let's go ahead and do some experiments. Why not? And he started doing these experiments, and he discovered certain laws and certain rules by taking into account certain features. He contrasted certain features, which were seed shape, the seed color of the pea plants, the flower color of the pea plants, the pod shape, the pod color, the flower position, and the stem height. But before we go into all this, let's talk about something more basic. The question really comes in, why did he do these experiments? So behind every experiment there is a specific logic, right? And these specific logic that we're talking about is right here in the idea that he found a very interesting thing happening when he started mixing traits together, like for example the stem height, alright? If he took the stem height and he took a tall plant, a tall pea plant, and he took a dwarf pea plant and he brought them together by the process of hybridization. What is hybridization? Hybridization is taking two varieties of plants, bringing them together, mating them, alright? And by mating them, what happens? They get a new variety. So what he did, say stem height, right? Stem height is denoted by capital T, and the capital T denotes the tall plants, like I have already mentioned out here. And the dwarf variety is denoted by small t. So he took a tall plant, he took a dwarf plant, he brought them together. But what did he get? He got all tall plants. So what happens? Capital T, small t. These give the gametes of capital T and these give the gametes of small t. And when they are brought together, he gets the capital T and a small t. Now, the deal out here is, though this is capital T small t, and today we know that it is capital T small t, but at that point he did not know that. And as a consequence, what happened was, he found that all these plants were tall. Why were they tall? There was no justification. For example, you have parents, right? All of us have parents. And everybody says that if the father is very tall and the mother is very short, the son or the daughter will be of medium height. But here that was not happening. That's how we look at things, but that's not exactly how it was happening out here. So it was confusing for him. So he started doing more experiments. So what did he do next? He took these tall plants and he interbred them. Alright, and he interbred them, and in that, what he did is, he crossed them, capital T into small t, capital T into small t. This is called selfing. This is known as what? It is known as selfing. By selfing, he got something very interesting. He got back the dwarf plant. Alright? So he got back the dwarf plant in the sense that he got back your dwarf plant by doing a simple cross, which we call as the Punnett square. Alright, this is known as a Punnett square. So I take the capital T, I take the small t, I take the capital T, I take the small t, right? And I cross it. So here I'm getting capital T, I'm getting capital T, small t, capital T, small t, and small t, small t. The interesting part here is this small t, small t. This is giving him a dwarf plant, whereas the rest of them are giving me tall plants. So he finds that in the second generation, this was known as the filial 2 generation, the dwarf plant suddenly comes back. Why does it come back? And that was the question that intrigued Gregor Mendel, and he continued with his experiments, and he did other ways of finding out the differences between the tall plants and the dwarf plants. And in doing so, he formulated the first law of dominance. Okay, first law of dominance. So he said that there must be certain factors. He used the term factors. He did not know the term gene at that point. He used the term factors. And factors, these factors are essential for a specific trait. The trait here is what? The trait here is being tall or being dwarf. The height of the plant is being determined by a trait, and that is the factor. So a factor is basically determining a trait. Alright, so a factor is determining a trait. So he says, alright, so it cannot be just one factor, but it has to be in pairs. Today we know that why it has to be in pairs, because all organisms that we know of, including us, we are diploid, means 2n. We'll go into that in another class, but basically you have to remember that we are 2n. So there will be 2 of each factor in every person, in every plant, every human being, every animal, mammal, so on and so forth, except a few organisms which are not. We'll talk about that in a different setting. So here, we need to remember that we are all diploid and capital T, small t. So we start with the factors in pairs for the 2 parents. So this is the parental generation. These are the 2 parent plants. And the parent plants are each giving a gamete, which is a gamete, remember, becomes always half in number. So only one t comes down, one small t comes down. So capital T, small t becomes the new genotype as we know it today of the new tall plant. Now the question really comes here is, why is it that when it's capital T, small t and there are 2 of each, why is it that they are tall? And this was the first law, which is the law of dominance. The law of dominance says if there are 2 factors and they exist simultaneously in a person, in an organism, and one of them is expressed while the other is not expressed, the one that is expressed is known as the dominant feature. What is not expressed, the small t, is called the recessive. So it's like this, like I always like to take this example of having 2 students, alright, a really fast student and a really thin student. So imagine you are standing and you're observing that there are 2 boys sitting on a bench, right? And there's this really fat boy and really thin boy sitting side by side. And then the fat boy decides to sit on the lap of the thin boy. Can you see the thin boy? Oh God. Because he has been completely removed from sight. But he's still there, he still exists, he's still there, suppressed by the fat boy, right? So this is a clear example of dominance, where the fat boy is dominating over the thin boy, and he is the only one that is being visible at that point. So this is exactly how dominance works. Dominance is the idea of what is visible physically, not what is there. What is there is called the genotype. What is visible is called the phenotype. So this is the first law of Mendel's, and this is known as the law of dominance. Alright? So the law of dominance was the first law that was given by Mendel, where he said that one feature was suppressed, one feature was expressed in a situation where both the factors were present, like capital T, small t, both are present. But then in the next selfing, we find that it comes out because of the feature of the selfing. Because the small t and small t will have a 1/4 possibility of being expressed in this new hybridization experiment. That is why we call this the first cross that Mendel does, and this is known as a monohybrid cross, and this gave rise to the first law of Mendel's, which is known as the law of